Okay, welcome all of you. My name is Ulf Söderlund, and I have the privilege to be sharing the remaining presentations during this morning session. And we have two presentations of world-leading experts. The talks should be about 35 minutes, followed by approximately 10 minutes with questions and discussions. Please make sure that your cell phones are in a silent mode. And with that said, I would like to welcome the first speaker, which is Professor Kliti Grice from the Curtin University at Perth. And her research specialty is to integrate molecular and isotopic information on biological material in order to reconstruct microbial and floral inhabitants, and especially during extinction events. And yeah, I don't have to read the title as well. So please welcome.
Thank you. Right. Good morning, everyone, distinguished guests, Crafoord laureates, ladies and gentlemen. So today I'm going to present the significance of microbes during mass extinction events and their role in exceptional fossil preservation. So this is using molecular geochemistry, so biomarkers and biomolecules and stable isotopes, together with what we learned about the geology of events through time. So I'd also like to thank the Royal Swedish Academy of Sciences and the Crafoord Foundation in London for this real privilege to be here, to be presenting to such a prestigious audience and such a prestigious event. So thank you so much. I also acknowledge my science team and international colleagues, both mentors and collaborators from around the world, and more than 40 PhD students who've had the pleasure of working with in the last 20 years. So this is just a very simplistic slide. We have a fossil prawn. You probably recognize that from the fossil and the prawn above. And on the right here, we have a molecule called cholesterol. Cholesterol is a biomarker which has the carbon skeleton representative of cholesterol, which occurs in algae. It's just lots of alcohol group and double bond. And this is what you find in the rock record under certain conditions. So the carbon-13 and carbon-12 and deuterium hydrogen composition of these molecules tell us about the biosynthesis, the source of these biomarkers, and the global water cycle as well. So today I'm going to discuss two of the big 5 extinction events in Earth's history, the cause and the recovery, and the ones I will talk about are the Permian and Cretaceous. So 252 million years ago, the Permian Triassic was when life on Earth nearly died completely. It was very bad. There was 96% of marine and 70% of terrestrial species wiped out. But it's also a really good time because it's opportunity for evolution to occur and recovery of life occurred. For example, this ichthyosaur evolved thoroughly in the Permian Triassic extinction. In Perth, in Western Australia, near where I live, there's the Perth Basin and there's rocks offering petroleum potential as well. And these were formed under very reducing conditions. So what caused the largest extinction event? As quoted by Irwin in 1994, it was a cascade of events. The Earth was significantly hotter. It was a global warming event. Lots of fire. The ocean was also very stagnant and devoid of oxygen. Massive volcanic activity took place because the supercontinent Pangaea was formed due to plate tectonics, and there was flood basalts reported in Siberia in the late 1990s attributed to this massive volcanic event. As Andy Knoll has mentioned, that there was high amounts of CO2 during this event, and there was also toxic levels of hydrogen sulfide in the oceans, which is some of our research where we've used biomarkers. So to start off, I just want to give you a summary of certain organisms called chlorinae, or green sulfur bacteria. This is the inland to Black Sea today. We have cyanobacteria living with oxygen. They make chlorophyll A and beta-carotene, whereas in these bottom waters, which are depleted of oxygen, you can find purple, green, and green-brown sulfur bacteria. These organisms use hydrogen sulfide as an electron donor as opposed to water in photosynthesis. And they need to capture longer wavelengths of light of the electromagnetic spectrum, so make very specific bacterial chlorophylls and carotenoids to do their photosynthesis. In the bottom waters here on the sediments, we get sulfate reduction and aerobically degrading organic matter to produce the hydrogen sulfide, which is utilised by these organisms above them. So these conditions are referred to as photoxonogenic. However, these organisms also occur in microbial mats, such as these smooth mat from Shark Bay in Western Australia, and I'll come back to that. So in the rock record, we've found biomarkers such as isorenaretane, just lost these double bonds here, from isorenaretane of green-brown sulfur bacteria, and bacterial chlorophylls C, D, and E shown here, which has a lipid side chain C15 farnesyl, and this B ring here gives rise to very specific low molecular weight polar compounds called malamides, which we can extract from organic material. So this is the supercontinent Pangaea during the Permian Triassic. We have no ice caps. The oceans were very sluggish and devoid of oxygen. We've looked at core material and outcrop material from around this supercontinent, and we find evidence of pigments of green sulfur bacteria at the extinction horizon and throughout the early Triassic. This tends to indicate that the hydrogen sulfide was prevalent in the upper oceans and light was available for photosynthesis by green sulfur bacteria. So we have killer seawater at this point, and we also have global warming, and we have the formation of the supercontinent Pangaea with volcanic activity. We've seen such conditions globally also for the end Devonian, the end Triassic, and the end Cretaceous extinctions. Now moving to the end Cretaceous extinction event. This was when a meteorite impact occurred, with 75% of plant and animal species becoming extinct, along with non-avian dinosaurs, reptiles, ammonites and rudous corals. And planktonic forams, nanofossils and land plants were also significantly affected. There was massive amounts of aerosols in the atmosphere, and light reduction and phytoplankton productivity declined in the oceans at this time. So this is the world 66 million years ago, when the non-avian dinosaurs were wiped out. The world very much like today, and a 180-kilometre wide crater was formed in the Yucatan Peninsula in the Gulf of Mexico. So this is just to wake you up a bit. So yeah, the asteroid was pretty big, equivalent energy of several million nuclear weapons detonating simultaneously. So in 2016, the International Ocean Drilling Programme and the European Consortium of Ocean Research Drilling actually drilled into the Chicxulub crater and retrieved core material from 506 to 1,335 metres below sea floor. This represents 48 to 66 million years. And this is the core material here with the KPG, the end Cretaceous extinction. Now, just going through some of these rocks, we have a granitic basement type rock, we have a scoopite layer, which is the tsunami layer above that. And then we have the post-impact sediments. So we've been looking at biomarkers. This is a PhD student, Bettina Schaefer here, in the crater core itself, through the whole core, from the basement through to the sediments, to look at evidence of transport of material or development of microbial communities within the crater, to establish how quickly photosynthetic life might have recovered. Do we see evidence of eugenic conditions when things became more stable? And given that the asteroid hit a carbonate platform, do we see evidence of microbial mats being flung up and thrown back into the crater? So the first set of results come from what we call tsunami markers. These are fairly new markers that we've been looking at in terms of the Chicxulub. Here we have perylene, which is a compound which comes from black pigments. Perylene quinone pigments are fungi which degrade wood in situ in sediments. And benzoylpyrene, which is a marker of fire or combustion. And when you look at perylene generally in sediments, increases in abundance with profile when it's formed in situ, such as this profile from a Holocene sequence from China. Now we look at the data of these two markers, perylene and benzoylpyrene, perylene being from our fungal wood degrader and benzoylpyrene from combustion. And this is within 1 day after the impact. This has been modelled by Sean Gulick, and this is the tsunami layer. What we see is an increase in perylene to benzoylpyrene at the height of the tsunami. And then when settling occurs within 2 to 3 years to 30 killion years, we see the reverse profile and we see species of the tsunami. So we actually, from a molecular level, are able to track the tsunami in the crater core, which was quite spectacular. So this was tsunami. And just to summarize the other biological biomarker information that we've obtained, this is shown on this slide. We had the impact, we had transport of debris, we have evidence of fire, but we also have evidence of microbial mats in the form of sulfur bacteria, but also in the form of intact lipids which come from filamentous bacteria which make heterocystic glycolipids. And these were prevalent through the profile. We also see evidence of cyanobacteria occurring within days to years to 30 killion years after impact. And then from 200 killion years to 4 million years, which is an extensive period of time, we see evidence of hydrogen sulfide in the sunlit zone of the crater water. So this is the tsunami. This is some of the data from various biomarkers showing presence of eugenic, but also microbial mat sources in the Chicxulub crater. Just briefly touching on the Triassic mass extinction, which occurred about 201 million years ago. This was a breakup of the supercontinent driven by the Central Atlantic magnetic provenance, yielding 2.4 10 to the 6 cubic meters of flood basalts. And it saw the extinction of the conodonts, followed by rapid expansion of the dinosaurs. The timing of the extinction has been a bit controversial, but we've been looking at samples from the St Andrews Bay in the UK. And just to summarise this data set without going through all of this data, we have an initial carbon isotope excursion here, and we've interpreted this. This is where no marine fossils are found. There's actually a shallowing of the water and the drying out period where microbial mats actually started to develop. And we see evidence of oxygenic mats, then eventually anoxic mats with purple, green and brown sulfur bacteria. And it's not until later, the second carbon isotope excursion, where we see the bicalcification crisis at this event, and driven by camp, and the presence of Fordyxonusinia. So we see a double whammy of the extinction interval higher up than previously thought, and this carbonisotopy excursion is attributed to microbial mats which are isotopically light. So this is just to say that these extinction events do provide us a key to the present. I've given you just a few examples of deep time, and you can see that in our modern day we have rise in CO2. We see very similar things happening in our oceans, acidification. We have bleaching of corals. We have dead zones being on our continental shelves and algal blooms. And these are some of the examples from deep time, particularly the Permian Triassic and Triassic Jurassic. So the second half of my talk is about fossil preservation. And you'll see the link with the first part of the talk. So we have normal preservation of fossils, where no material, original material is completely replaced with minerals. We can have selective preservation, where certain biopolymers, such as plant cuticles, actually survive degradation and are actually preserved. And we can have exceptional fossil preservation, where original material avoids full decomposition, such that there might be soft tissue, but you also might find biomolecules intact, such as cholesterol itself, not the biomarker cholesterol. So we've been working on carbonate concretions, which are mineral aggregates from a few centimetres to metres in size, comprised of siderite, iron carbonate, calcite, apatite or dolomite. Many of these do contain exceptionally well-preserved fossils, and this provides us insight to extinct organisms, evolutionary information and paleoenvironmental depositional information. Fossils can preserve organic matter and contain biomarkers, but there's the potential of finding intact sterols in very old samples, along with things like fatty acids. So I'm going to show you where we've got some exceptionally preserved fossils in Australia. These come from Perth here, where Curtin University is. Here is the Canning Basin, the Leonard Shelf, a massive reef complex of Devonian age. And this is where Australia's Latrostat occur. These are some examples of both bone and muscle tissue in 3D preservation. This is a go-go fish. We have original bone with histological detail, and muscle which has been mineralised and preserved as apatite. This is the work of Kate Tronistick, John Long and Derek Briggs. So these are the nodules that occur, or the concretions at this location. They're from the reef building area in the Canning Basin, from Gavetian to Enfamanian. The concretions are Frasian age, and these weather out of the basinal muds. So we've been working on biomarkers in fossils in terms of how what that can tell us about preservation of soft tissue, and can we use biomarkers to say something about the organism itself, actually identify it. So this is a concretion from the Canning Basin which contains an invertebrate called Muschia. It's a bit of a mess. I think that's where it gets its name from. These paleontologists don't know what it is, other than it's an invertebrate. And this has been taken, the fossil layer separated from the matrix layers, so different matrix layers here and the fossil layer, and extracted for biomarkers. You can see from these GCMS fingerprints, this is the chemical analysis, that the matrix, the calcium carbonate, is somewhat different to the fossil matrix, fossil layer here. And you see that the matrix is really comprised of long chain alkanes, whereas there's lots of nice goodies in here in terms of biomarkers. So what did we find in this fossil? Well, our famous isorenaretane, a biomarker of green sulfur bacteria, was only present in the fossil. It wasn't present in the concretion surrounding it. The concretion was largely comprised of long-chain alkanes with very depleted carbon signals, which are attributed to sulfate-reducing bacteria. What we can also find in this fossil, just in the fossil sample, not in the matrix, are the abundance of steroids, such as C27 cholesterol, which comes from cholesterol. And the percentage of the steroids in this example here are concordant with a crustacean. There's no shell so it's not a mollusk, but the percentage of steroids are consistent with this being identified as a crustacean. So there's our prawn. So what happened with this organism was that it was happily feeding on phytoplankton in the Devonian shallow sea. It probably died because of toxic hydrogen sulfide, a little bit like the Permian-Triassic extinction and the Triassic-Jurassic. It was the carcass was probably recycled by anaerobic photosynthetic bacteria by building a biofilm around the carcass. And even though hydrogen sulfide maybe killed the organism, hydrogen sulfide actually has the can actually link with molecules and form carbon-sulfur bonds, and that was actually the case. We found carbon-sulfur linked biomolecules in the soft tissue as well. And then the organism sank. This might only been 0.5 metre, very shallow water column, very quick, probably preservation occurred, and the sulfate-reducing bacteria, which produced the H2S in the first place, built a calcium carbonate concretion around the decaying carcass as the alkalinity dropped. So this is what we found in these samples. I should also mention that we also found 70 different steroids in the soft tissue, which were all the diagenetic products of cholesterol, including cholesterol itself, only in the soft tissue. So we were actually able to capture the transformation of sterol in the water column here in Geosciences, actually in the soft tissue and the fossil material. The next example I'll give you is from a vertebrate bone of an ichthyosaur. This is Jurassic concretion from the Posidonia shell, and this is the work of Chloe Platt. Here we have a pure calcite body, we have a calcite, pyrite and traces of quartz rim, and we have the nice bone here. This was subjected to, unfortunately, destructive analysis. However, the imaging of this bone from XRF mapping, phosphorus mapping, shows a corticular hard bone and a trabecular bone. And this trabecular spongy bone is filled in with sparicalcite and barite. When the sample was treated with lytosikic acid, we were able to see flora-apatite remains, encrusted collagen fibres in this 183 million-year-old bone, along with a series of red blood cells, which are 2 micrometers in diameter, and I'll come back to why they're quite small. We also found in the bone intact sterols, not the biomarker, but cholesterol itself in this 183 million-year-old bone, along with ethyl cholesterol, which is the algal sterol from the same environment. And the isotopic composition difference between these is due to the trophic effect, the ichthyosaur being higher up in the food chain, obviously, than the phytoplankton. We also find only in the bone the entire diagenetic continuum of cholesterol and all the intermediates as well. So really, really well preserved. And green sulfur bacteria. So we have a similar preservation as in the Devonian concretion. So the life of the ichthyosaur, well, you know, it evolved from the Permian-Triassic extinction. The oxygen levels in the ocean were very low throughout its lifetime. We have the diet, cholesterol, and the isotopic composition of the dietary cholesterol intact in this 183 million-year-old bone. we find a green sulfur bacteria, which is probably providing the EPS or the glue to actually help to preserve this material. We see collagen fibers and small red blood cells. These red blood cells are probably smaller than modern. There's obviously some change in the size of the red blood cells due to shrinkage. However, if you look at animals which live at high altitude today, they actually make smaller red blood cells to efficiently transport oxygen around the body and increase the surface area to volume ratio. So we think that this is why in the ichthyosaur, this was very small red blood cells. We did also find these breakdown products. However, this could be from heme, but it's also could be from chlorophyll, and the isotopic composition actually helps us to determine if this breakdown product of heme is actually from the hemoglobin itself, and we think that is the case. So this is our ichthyosaur at that time, well preserved in concretion. The other sorts of concretions, and I don't have results for this just yet, this is still to be published, but the Mason Creek is an area we've been working on as well. Mason Creek is from the Carboniferous, and the way that these concretions are formed are somewhat different to the concretions from the Devonian, and this is probably driven by decomposition of biopolymers like leaf cuticles, breaking down to acids, reacting with iron, and increase in pH leads to iron carbonate, and the concretion growth probably occurs with increasing concretion diameter, so it grows outwards like this, and that allows anoxia before significant decay of soft tissue. So the ones that we're working on right now, I hope to get some results next week from the Tully monster. The Tully monster is from this locality. It's the state fossil emblem of Chicago. It is inside one of these siderite concretions, and it's been recently very controversial because it was thought to be an invertebrate, but now they've actually found cartilage type material. They think that it's got an eye bar, it's got eyes. They find breakdown products of the melanosomes of the eye in these samples, and it's also got teeth, and we're actually going to try and establish the diet of the Tully monster, which will add significant information about this organism. So my current project, which is an ARC laureate fellowship from the ARC, is for 5 years, and it's just at the beginning. We're looking at concretion material from around the world, from different geological timeframes, to actually look at new biological material, particularly intact biomolecules for the first time, because we think that these are so rapidly encapsulated that preservation probably occurs within a few days to months. So this is a fabulous opportunity and an untapped resource of molecular information. What we're also doing is I have a biocement expert who's an engineer who is combining biological cement expertise with microbial mats. We know microbial mats play a key role in fossil preservation, and we're actually mimicking different environmental settings and looking at how in situ incubation experiments with different tissue, who's there and what they're doing, and what the environment was like to help understand how preservation of this material occurs. And I'd just like to finish there with this quote from Charles Darwin: "It's not necessarily the strongest of the species that survives, nor the most intelligent, but the most responsive to change." And that's something I hope the politicians actually learn from our understanding of climate change today. And thank you for your attention. I've also been to Spitsbergen, and I sampled the PT boundary there, so I also agree it's a fabulous place to do science and geology, and I'm a chemist, so I was out there as a geologist, field geologist, or trying to be, and I've had fabulous trips around the world, and yes, some nice pictures there. So thank you for listening.